we have atom, atom number, then atom name, like here NCCO, so nitrogen, carbon, carbon, oxygen, nitrogen, etc. Then that atom, which residue does it belong to, glycine or valine, lysine, etc. Then which chain is it in? So if since it's a dimer, so we have two chains, chain A and chain B. Then the residue number, glycine 137. So you can see the information here, and these are information like x, y, z coordinate. So if I open the, yeah, so as I said, it has a header which contains the metadata about the PDB files. It often includes information about the experiment used to determine the structures, such as X-ray crystallography or NMR spectroscopy. Then we have compound and helix records, as you saw. Then heta-atom records, which contains the metal ions, water molecules, ligands information. Yeah. So for atom records, we have atom type. Yeah. So atom type. Then we have residue name. This is the residue name. Then we have chain identifier, A chain. Then we have residue sequence number 137. Then XYZ coordinate, X, Y, Z coordinate. So whenever you have a protein, you basically enclose it in a box, in an XYZ box, in a cuboidal box with a specific origin. So based on that origin, you can tell that this atom is present at this XYZ coordinate with respect to the origin. And this 1 and this 68.38, this number corresponds to the occupancy and the temperature factor. So occupancy is, this indicates the fraction of molecule in the crystal that contains the atom at given position, and the temperature factor, which indicates the mobility of the atom. So yeah, and one more thing, like you can see the names of the atoms are very different here, like N, Ca, Co, then you have Cg, Cb, Cd1, Cd2, but at the end of the day, these are just carbon. So why do they have different names? Because when you have different atoms of the same atoms in the residue, you would like to name each of the atoms separately so that you can identify or tell which atom is which atom we are referring to. So by convention, people have set some convention names so that they can put Cb, Cg, Cd1, Cd2. So there is a convention that is followed. So you can look up in the net and like how this actually works. Okay. So then there is connectivity records at the bottom. So yeah, these are the connectivity records. This connectivity section includes the record such as which describes the bonding between atoms in the structure, particularly important for non-standard residues and ligands. So this section basically helps in molecular interactions and chemical environment within the structure. Okay. So I hope this is the general PDB format that we usually see, and it's very simple and very clear. So the most important part is the understanding how the atoms are there, the heta atoms and the other atoms. Okay. Yeah, this part. Okay, so let's move on. So the next most important type of file structure that we usually see is the PQR file. So PQR represents, P represents position, which means X, Y and Z coordinate. Q represents charge and R represents the radius. So this file is very important because various softwares, when you input PDB files, it takes a long time to calculate the potentials or whatever values we are calculating. So we usually give PQR files, which is just like a consolidated data that is needed, and it contains some more information that might be missing in the PDB files, like some atoms. So yeah, so the PDB file does not contain metadata, and it contains information readily usable, similar to section 4 of PDB file. So yeah, it contains this atom records only most of the times, and yeah, P gives the XYZ coordinates, Q gives charge, and R gives the radius. So I will show a PDB file. Do I have a PQR file? Yes. Then yeah. So this is a PQR file. You can see it only has the atom type, the atom number, the same atom namings like NCAC, then glycine, chain, residue number. This is the same as what we had in the PDB file, and these are X coordinate, Y coordinate, Z coordinates. The PQR, so this section is P, this section is Q, and this is R. So Q is basically the charge, and R, it's in Armstrongs, which is the radius of the atom. Okay, so this file is just completely full of all this. It has nothing more and nothing less. I'll tell you how these PQR files are generated in the later sections. Okay, so the next thing is XYZ format. This is just very simple. It has the atom name, X, Y, Z coordinate, that's it. So it's very straightforward. And the DX file, I'll show the DX file because later on we'll see the potential using APBS coming in a DX format. So it's better to understand now so that we can look at later on and don't get confused what is what. So I have mentioned this link, so you can just go here, APBS, what is this? Okay, yes. Yeah. So there is this link I have mentioned about for understanding .dx file structure, refer this. So you can copy this link and open it in your Safari, Google, whichever browser you are using. Oh, very nice. What's going on? Yeah. Okay, so have you opened this link? How many of you have opened this link so far? Okay, so I'll wait a couple of minutes. Everyone please open this link so that I can show the DX formatted file and we can compare what's going on with this format that they have mentioned. Okay, so I'll continue now. Okay. Can you speak louder? Can someone give a mic? Or you want a brief introduction of what all has happened so far? Okay, so nothing much I have done. It's just I've explained what are PDB files, how do they look like, PQR files, and we are just discussing the DX format, and yeah, that's it. Nothing deep has happened yet. So yeah, just open this link. You'll open this link. Go to this AIIM workshop, and you will find all the PDB files and codes that I'll be referring in this workshop present. Okay. So I hope everyone has opened this link. Now the DX format, when you pass the PQR file, the input file in APBS through command line, you get a file like this, basically. But you can't tell what each of these columns and why only 333 are there, what the other things mean. So the format is mentioned here. So I'll just better. So the first row represents the size of the cuboid that we are enclosing our protein inside. So NX, NY, NZ. So NX, NY, NZ basically represents the X, Y, and Z size of the cuboid that we are using to enclose our protein. Then we have the origin mentioned. Then okay, so let's start from this part. So here the DX file has the potential that that is calculated at each grid point. So since we have 30, how many do we have? 353 into 193 into 257. So these many grid points, and at each grid point the potential is calculated. So we have the potential at 0, 0, 0, this number, then the potential at 0, 0, 1, this number, then the potential at 0, 0, 2, this number, and so on. So this goes on and on till the 0, 0 ends, 0, 0 nz is over, and then 0, 1, 0 starts. Okay, so it's a very ugly and very non-intuitive file structure, but it is what it is. Okay, now let's start with the PyMol UI part. So open PyMol, everyone, with the, let's see, with 7y6i, 7y6i.pdb. So this is a PDB of NCC, which we in our team we used. Okay. So go to PyMol, file, open. Very beautiful. Okay, so have everyone opened this? Say what? Yeah, download the PDB file and open it in PyMol. How many of you have opened it? Back row. Okay, here. I'll wait 2 more minutes. So you can just use your mouse pad and scroll around, see different parts of this protein. You can see there are some colored spheres here, which I'll tell about, some green spheres, some purple spheres, and then helixes, then these are chains of lipids. So this is the whole protein structure. This is a very complex protein structure having around 1000 something residues. Okay. Okay. So when you go to display section above, you can go to sequence, just click sequence, and you can see the sequence of each residue that is mentioned here. So starting from one residue, that is glycine, that we saw, and you can see it's okay. The font is small, right? Oh, it's not getting zoomed. Okay, anyways. So see in your laptop, the first residue here is like G, if you can see, GV, right? GWV. Okay. So how we operate in PyMol? Just use a mouse pad and move around, do whatever you want to do, and okay. So but if you don't remember each letters symbols, you can just go display, sequence mode, residue names. So this will give the 3-letter words, so glycine, valine, lysine. So this is much more intuitive, and you can see which residue is which number. Okay, is it clear so far? Okay. So yeah, so let's see, displaying sequences and coloring. Okay. So how do we know that this protein, this residues, is present in which part of the structure? Because this structure is a ribbon structure, you don't know like where the glycine is. So if you tap on this glycine, some part got highlighted. Where is it? Okay, I don't see. Let's select more. Yeah, this part. If you see some dots, some reddish dots come here, so you can tell that the this glycine and valine and the other residue are in this part of the protein. So but you cannot tell like this is present in the space somewhere, right? It's present somewhere in the space. So to see the residue, when you select some group of residues or some selection, this cell variable comes about, and you can just go action, rename to some better name than select. Let's do uh, part 1. Okay, so the part 1 got renamed, and we can just go show, and we can show this this selection in various forms like ribbon, spheres, sticks, surface. So let's go with sticks. There is sticks. So if you can see, some compounds that you usually see in books came here. So, and but it's the same color, right? So we can just go to C, color. Let's go for red, and yeah. So now it's much easily visible. So you can see glycine, valine, and I think this is tryptophan. Yeah. So all this got highlighted, and now you know how to color them and how you can see which part of this protein looks like what. Okay. Similarly, if you can just take the whole protein and color the whole thing, or maybe you can just go show and you can see the surface. Okay, this is not surface, this is spheres. Yeah, this is the spherical representation of the whole protein. You can go to H and just hide it, hide everything. Okay, no, not everything. Show everything. Okay, whatever. Let's open it again. Yeah, okay. The color changed. Okay. So I hope displaying sequences and coloring is clear. And now let's see the alignment between two proteins. So whenever you are seeing different conformations of the same protein, you want to compare them, like how these two proteins are different. So that's where alignment comes in. So alignment basically tries to match the two proteins as best as possible. So you can do this. Let's take two proteins I mentioned here, this 7Y6I and this 8FHP. Download these two proteins and open it in the PyMol in the same place. So you can just go for, wait, where is PyMol? Yeah. Just delete this object. Okay. File, open, yeah, 8FHP. So 8FHP is, now you can tell that it sort of looks like the old structure, but you would like to know how is this exactly matching, or is it like from where, which part of the protein is it different from? So you want to align them so that it's easier to compare. Right now there are some shifting and translation, so it's not exactly aligned. So what you do is go to actions, A here, go to align to molecule 7by6i, and what, invalid name, align molecule. Okay, weird. Let's try super. No, it usually happens. Wait, I'll restart. Delete. Okay. Yeah. So they came in different colors now. So let's try to align it. Align to molecule 7by6i. Yeah, done. Okay. So if you can see, the representation is much better. Okay, I'll change the coloring, or maybe not. Okay. So also you see these yellow lines. Can anyone guess what these yellow lines like this mean? Said what? Loop? Can you speak in mic so that everyone can hear? Yeah, this yellow line. What do you think this yellow line means?
Those are bonds maybe.
Bonds, but we are just taking two proteins and aligning them. We are not having any. Yes. So for the same residue, one residue of this protein is is present here in the other protein. We are not having any reactions that their bonds are forming. We're just seeing visually how these proteins align and how they look like. So the one residue of this protein is basically mapped this to this protein. Like it's present here. So similarly you can see this protein is present here, this protein is present here. Some of them may be overlapping perfectly some part of the protein because this is a very complex protein. So we can just delete that part or just hide it because it's ugly. Yeah, okay, so it's now much better, right? Okay, so let's scroll through it, and so most of it is very same, it's just shifted a bit. But one thing I would like to point out is these things. NaCl, NaCl. So in this NCC protein there is something special that since this is a part of an ion channel, this protein, we have sodium and chlorine present in this. Okay, I'll just hide this one. Okay, we have sodium and chlorine present in the molecule. So you can see that the purple spheres are Na and the green spheres are the chlorine. And we have other these chains whose names are also written here, Y01, Y01. So if you want to know their exact name, you can just go to the PDB file and it should be mentioned somewhere. Okay, I'll just search Y01. Yeah, where is their names? Okay, see, so this is the name of that Y01 cholesterol hemisuccinate, whatever. So yeah, okay, so now that we have aligned it, I'll just delete this also. Okay, so the alignment part is done. Now one thing that commonly we want to know is, since this is a part of ion channel and these are two conformations, we want to know how the ion channel exactly opens up, how it closes, get a rough idea of what's going on. So then there is something called morphing that comes. So morphing is basically like an animation between one conformation to other with the possible route that it moves with. Okay, so how do you morph? There are also various morphing algorithms also, but you can look it up on Google, and I'll not be telling about the different morphing algorithms. So you can do action generate morph to molecules 8FHP. Oh boy. Sorry, what? We are morphing one conformation to the other. So we are morphing 7/6i to 8FHP. Yeah, two molecules. So go to generate, generate morph to molecule 7/6i. Okay, it's showing some errors. Why? First, let's align them. Then morph them. Yeah, so morphing takes a bit of time. So before morphing you must align those two proteins, because we don't want unnecessary motions that are also present, that will also come in this morphing part. We just want to know that from one conformation when they are aligned, how much difference does it make. Okay, so it takes a bit of time. Okay, I'm done. So the morphing is basically like usual animations. We have a set of frames and you just run them in a speed so that it looks like an animation. So if you can see here at the bottom right, we have 1/30 written here. So what this means is this is the first frame of that morphing, the animation thing. Then as we move the arrow, we can see the different motions that are happening in this morphing. Okay. So if you want to see an animation, we can just go like this. You can just press this button here and it will go like this. So now time to analyze. So we took two conformation of proteins, 7/6i and 8FHP. Both are part of ion channels, and our goal is to see maybe how the ion channel opens up, how it closes. So if you can see, there is a different sort of motion of happening around here. So basically the ion channels kind of opens and closes and opens. So if we compare not just these two but maybe more, we can see the whole ion channel closing and opening. So I have just taken two. So it's a very good visual of how the ion channel may be moving. So maybe the sodium ion moves somewhere here. Okay, there is a sodium ion here. Let's just see that. So you can see some motion of ion channel, this sodium. So of course in between also there may be some other states, but this is just a good approximation of maybe how the ion is moving around. Cool, right? Okay. Okay, let's stop this. Yeah. So the morphing part is done. I hope things are clear, very simple. Is there anyone who couldn't do this much? What happened? Okay, I need mic. Can someone give mic? After aligning, how did I morph?
Yeah. Could you just
Okay, so you just go here, action,
yeah
generate,
generate?
morph,
Okay.
and then molecule. 7/6.
Mhm.
It crashes. That's, I think that's software
Yeah,
or laptop issue.
not
I
from
cannot
other
help with that.
7.
Okay, things are clear so far? Yeah, I'll take that as a yes. Okay then. So the next part we would like to understand is mutations. So various times it happens that the proteins have various mutations for diseases or some syndromes. For example, the lysine got changed to cysteine. So for smaller molecules, for smaller proteins with 10, 11 residues, it may happen that when you change a mutation, the whole protein structure may change because it's already very small. Each protein has a very important role. But for larger complexes like this with 1,000 plus, it depends on where the mutation happens. If let's say I take a where is it? Yeah, if I let's say take some random residue here, and since we are analyzing ion channel, there is nothing going on in near that protein which is being mutated, so it would have very less effects on the observation and the calculation that we would like at the end. But if we take a residue near the sodium and see if that's mutated and that's causing all the changes, all the syndrome that's happening, then it would be great to analyze and then calculate the values based on that. So whenever you are mutating, you can always create the PDB file, the new PDB file, and use that PDB file as a new protein and then do the calculations. So the electrostatic calculations will be done in the second part, but so far let's understand how do we do mutations. Okay, I'll just delete this morph because we don't need it anymore. Okay, so in this protein, we go to wizard. Wizard has its mutagenesis. For mutagenesis, since we are dealing with proteins, we go for protein. Okay, so now it opens up a lot of options. So two things: we have to select which protein we would like, which residue we would like to mutate and to what. And then there comes rotamers that we have to select, which I'll tell. Okay, so let's go for 475. 475 is serine. Why did I choose 475? Because as we were doing, so there is a list of mutations that happens in Gitterman syndrome, which we were dealing with, and there is a big list of the mutations. So we are just taking one of them, which is 475 cysteine. So we are currently it's what is it? Yeah, currently at 475 you can see it's serine, but we would like to change it to cysteine. So we 7 by 6 I. Okay. So we select this residue. Now we would like to change it. So go to new mutations, and we would like to change it to cysteine. Okay. So now when we change it to cysteine, we get various possible rotamers. So where is that? Okay. So can you see this 1/3 here? So these are basically different possible rotamers. So after selecting serine, go to no mutation written here, change it to cysteine. Okay. So when you see the next possible rotamers, oops, yeah. Okay. So there is a percentage written here, and you can see the strain which is written there. For each rotamer we have a different strain. So there is in pymol the maximum percentage of occurrence that's possible. But this section, oh, so I just went to wizard here at the top, mutagenesis protein. Then you go to new mutations, select the residue, go to new mutations written here, and just select the select the new residue that you want to mutate to. Okay. So sir, what? Change it to cysteine. Okay, so I was explaining that there are different strains and different percentage of occurrence there. So currently for the first rotamer, we have 73.3% occurrence, but it has a strain of 35.52. So usually it happens that pymol chooses the one with the least strain, but that may not be the one with the maximum percentage of occurrence. So what we want in calculations that we use is the most occurrence one, like that is the first frame. The first frame will always have the maximum percentage of occurrence, but it may not have the least strain. The least strain will be automatically chosen. It may be 8 out of 21. There can be many more rotamers. So for usual calculations, what everyone takes is the one with the most percentage of occurrence rather than the least strain. Coincidentally, here both of them match, so it's great. Yeah. Sorry, what? What is? Yeah. So that's what I'm telling. So you have to apply here, but which rotamer we have to choose, that I was telling. So you have to choose the first one with the most occurrence, percentage of occurrence here, rather than the least strain. You can also choose that, but it's like the convention that is followed. So you take the, okay, I'll show how the rotamers different look like. Where is that? Okay, I lost it. where is the selection? Oh, there, somewhere there. Okay, fine, found it. What? Yeah. Okay, so I'll show why most the least strain and the most percentage occurrence have the effect on this protein. So if you can see this, this is the protein that we this not the protein, the residue that we have chosen, which is present in a stick form, and you can also see these red cylinders. So these red cylinders show the different these are the bumps or the collisions, the restraints that happen with the other residues. Okay, great. Red and okay, better. Okay, or rather I'll change it to green. Okay, so now when I change the different rotamers, you can see the cylinders also change. See, this has a lot of red cylinders, lot of bumps, so this is very strainy. Like you can see 51.95 strain is there, so it's very high. So that's why we don't choose. And for the other ones, this has very like only one and like very comfortably fit in here. So that's why we will choose the first rotamer. Okay. So we just click apply here and we are done. So the residue is now changed. So you can see at 475 position here, the serine is changed to cysteine now. Is everything clear so far? Okay. So I want to highlight one more point, is that for large proteins, as I said, point mutations don't take a lot of difference, make a lot of difference in the structure. So pymol for unit mutations give almost the correct structure, but when it comes to smaller proteins and or we are changing a large number of residues, then it's better to use AlphaFold or some other protein making softwares, building softwares, to get the new structure. But for point mutations for larger structure, larger proteins, pymol doesn't give a big effect on the structure. Okay, so now we come to part 2, which is the Python and the pymol. How we use Python to automate our work, because we are not going to do everything manually, and if you are using 100 proteins at once, or as I said 30 frames of morph at once, you are not going to do it manually at the same time. So that's where Python comes in, and you can just write a Python code and go to pymol command line, which is this place here. This is the Python command line, and you use run pymol code.py. So let's take an example of a Python code. So one thing to note, you don't run this Python code inside your VS Code terminal or your terminal. You run this inside the pymol. If you run it here, it won't work. Okay. So this is the same thing that we were doing, but for but in pymol. Okay. So sorry, what? First. Okay, fine. So we select the mutant. So mutant is a new residue that we want to change. So I have written argine here. Let's put it to cysteine. It should be a 3-letter code. Then we select the number that we want to choose, like the which residue number, which is 475. Then pymol has command cmd, which we use. So command.wizard mutagenesis opens that same panel that we usually see. Then command.getWizard set mode mutant, and then now we select which chain and which residue that we want to use. So do select from get wizard we are creating this object which will select the chain A and residue 475. Then again, as I said, we want to get the first frame which has the maximum occurrence. So we go for command dot frame 1 and then we apply it. So this code is mentioned in the given in the repository, so you can just see it, copy it, download it, and try to run it on your own. So command dot wizard, we just apply and remove the panel and then save it. Okay, so this is a very easy Python code. You can also loop it for various files and maybe more operations if you would like to do. Okay, so let's do it here. I'll delete this. Sorry, what? Okay, so if you can just run the Python code with the protein open, it will give the same effect that you had previously. To hurry up, I'll not run it right now, so we'll just skip to the next part. So here are some common commands that you want to use. Acha, by the way, you can use this command dot fetch protein directly in your PyMol command line also instead of running it in using Python. I'll just give a brief example. Command dot fetch 7x6i dot pdb. Does this work? Or just fetch? Sorry. No, I think this also works. Okay. Anyways, so these are some of the common things that you usually use: aligning, getting the number of states of a morph, setting the name, deleting, creating selections. Then again, as I said, this is the same code as we used for our mutagenesis. Okay, now let's go to electrostatics, APBS electrostatics. APBS stands for Adapted Poisson-Boltzmann Solver. So it's usually used to calculate all the electrostatic values, the gradients, the potentials of the protein at each grid points. And because when you are having various force field, when you want to know how the ions move, you would like to get some values to check and verify. So this is APBS electrostatics come in that place. So we have two places. Either we can go to web servers. So for web servers, we have the APBS web server, where you can just write APBS web server, and this website will come at the top. So you just get started with PDB to PQR. Now this web servers follow exact same thing as we do in our command line, and this web server has the best part. Like if you are not able to understand when you are using command line which command to use, like which parameters to parse, you can just go CLI. It's written here, force field parse, and this one. So the best part of this thing is you can get the CLI representation also of what to add and what not to add. So when you go to upload a file, select file, let's say 7x6i, upload. Yeah, we can search it directly, but if you have any proteins, you can just upload it directly. You can also search it and just start job. You can select various parameters if you want to give the different pH. Do you want pK calculations or not? And each of them has a different command line parameter that you can parse. So we'll use this and then start job. Okay, so it will take some time and we'll not wait here. Till then, there is also a Delphi server, which is also very good. It gives you the the gradients also along with the potentials. And here you can select more things. Like for example, the dielectric constants, the solve concentrations, and there are various more iterations that you would like the command to move in, and specify force field, specify pH. So it does the same thing but better. Okay, so this is how you use the web servers. Then we have the command line tool usages. We'll do this right now. This is used for automating your work and not waiting a lot for web servers, because web servers take a long more time compared to our command line. Okay. Yeah, I just explained this part. So let's try getting a PQR file from our PDB. Oops. So So let's take this command I have mentioned here, the second one. You can just copy this command, write it in your terminal. So how I have written this is, when you write PDB to PQR, it gives, and like PDB to PQR help, it will give a lot of options, but you don't know what options to choose. So just go to the web server and you can choose what all do you want. So sorry, what? The second one, this second code, this for better example case one, this one. Just copy this in your command line. Okay. So yeah, if you see, the job is done, and we are going to generate exact these files, but not this random name. I will name this. Exact these files will be generated in your command line. So okay, as I explained, you can just see which CLI you want to give the command. So I have these various parameters. Titration state method is propagate. We are passing the pH as 7. We are dropping water. This is for beautification, keeping the chain in the PQR file. And this is very important. We want this because when you are giving APBS command line tool, we don't pass the PQR file as it is. We give a protein.in file, which is of some specific format, and generating that, writing that .input file is a bit complicated. So PDB to PQR automatically generates this input file. So just run this. Also, yeah, give the give your PDB file 7y6i, and we'll name this as 7y6i.pqr. Sorry, what? That will be given the protein.in name. Okay, so we can also have right here 7y6i. Okay, this will run the job very fast, and you are almost done. Right? Yes. So all these informations are also given here. You may want to read this, or you may just skip it. Okay, so now we see that this .input file is made. Let's read this. Okay, so we are reading molecule PQR file, which is 7y6i.pqr, and so .input file has 3 parts: read part, and the print part, and this electric part, whatever you say, which has all the parameters that you want to pass through. So you already have a pre-made box. You don't have to assume what the length is, the best length is. This is the dielectric constant, and you can specify the pH here somewhere also. You can just go to the website and sorry, what? Sorry, I'm not able to hear. Yeah. By default, it's water. So this file is made, and you can also see the PQR file is also made. This 7y6i.pqr file, you can also see this. Then now you want to run APBS calculations. APBS calculations can be done very easily. Just do write APBS. We generated the input file, the 7y6i.in. this will do all the calculations with different iterations, and you can see what all values it takes: solute dielectrics, solvent dielectrics, temperatures, what other electrostatic energy is also written here, the total energy. So this values are also very important, so don't skip this. And okay, so now this dx file came, right? So as I said, the APBS generates this .dx file that I mentioned in the previous part. So when you open this 6i, it looks exactly like what we saw before, this dx file. It contains all the potential at each grid point of the box that these have chosen. So the box size is 193 times 225 times 225. Okay, so now we have generated this APBS software, and then we are done. So that's it for my side. And so in this we covered the electrostatics part, and this, yeah, go on.
You can ask any questions related to this after the session is done.
Yeah, I hope I didn't take too much time.
No. Now Sushmit will continue with the rest of the workshop.
Hello, am I audible? Okay, so I will be speaking about an important part of computational biology, which is molecular simulations. So before that, I would like to start with some statistical physics. How familiar are you guys with stat physics? So if you are familiar, then I can go first. So StatMech is basically assigning probability, doing probability theory with systems of large, like you have large molecular assemblies, and you do statistical and probability theory with that. This is like a bridge between the microscopic Newton's laws and the macroscopic thermodynamics. So you need to know what a microstate and a macrostate is. A microstate is like the configuration of the system, which the microscopic configuration of the system which the system is in. So for an ideal gas, it can be the set of all momenta and positions of the gas particles. And macrostate of a system is the large-scale thermodynamic properties, for example, temperature and pressure of the system, which are seen macroscopically. So for each macrostate, you can have many possible microstates, and you do statistical mechanics on that. So before that, I need to talk about what a phase space is. So phase space is the space which contains all the configurations possible in the system. So your system is a point in the phase space, and because it takes one configuration. And this system with time moves in the phase space. So it changes its configuration with time. What an ensemble is it is a large number of copies of the same system with different initial conditions. So they form a distribution in the phase space. So you will have many points in the phase space, each of them representing a system. And this distribution can be parameterized by a distribution function. So this gives you the density of the number of points at each point in the phase space. So that I am calling it FPQT. Now if you have this distribution function and your system follows Hamiltonian dynamics, you can show that this equation called the Liouville equation is valid. And this states that the phase space, this distribution function doesn't change its density as you move along with the trajectory of any particle in the system. And this is called the Poisson bracket, which is defined in this way. So the Poisson bracket of f and h plus the del f del t is 0, which is the Liouville's equation. So I'll talk about ergodicity. So ergodicity expresses the fact that a point in the phase space is a system can access all the points in the phase space. So if all configuration in this phase space is accessible to a system in some amount of time, you call the system as ergodic. So if a system is ergodic, you can talk about probabilities of finding the system at a region of phase space, and that you can calculate by the amount of time spent in that region divided by the total time as the time goes to as you measure for a long time. Also, you can find that probability by multiplying the phase space density times the volume in which you are interested. So if you have these two ways of calculating probabilities, you will have two ways of averaging quantities. So first way is doing the time averaging, where you measure the the variation of a quantity with time, which I call L. L is any observable, and you divide that with T, and you observe over a long time, and this is time averaging. And you can also do ensemble averaging. So here you multiply it with the distribution function and just do averaging. So the ergodic hypothesis says that these two kinds of averaging are equivalent when you have an ergodic system. So most of the time in molecular simulations, you assume that your system is ergodic and you assume that this is valid. Now, if you have a system in statistical equilibrium, by statistical equilibrium I mean that all your macroscopic quantities on average are not changing, so they remain constant at time. This means that the distribution function also won't be changing with time. That means your distribution function doesn't explicitly depend on time. So from the Liouville's equation you'll get this part is 0, which means that your distribution function is only a function of your Hamiltonian, which I have told here. So you can have different types of ensembles possible, and I'll list down some of them here. So one of them is the microcanonical ensemble, in which you have constant number, constant volume, and constant energy. And in this we have to assume a crucial fact, which is called the postulate of equal a priori probability, in which you say for these macroscopic constants n, v, and e, all the microstates corresponding to this macrostate are all equally probable. And this is intuitive because your system is ergodic, but there's no proof of this. So if you assume this fact, you can assign a probability distribution to each of this microstate, which I have told here. So when your Hamiltonian is equal to E, all of them have equal probability. The next ensemble is the canonical ensemble, where you have constant number, constant volume, and constant temperature. How do you maintain constant temperature? You connect it to a heat bath, and that maintains the constant temperature. So if you do all the formalism in StatMech, ensemble, which is the famous the Boltzmann distribution, and which says that your probability of a microstate is proportional to e power minus h by kt. And this z is the normalizing factor, which is called the canonical partition function, and it can be calculating by summing all those e power minus h by kt. And this is the most important parameter which you can achieve from this canonical ensemble, as all the thermodynamic quantities are encoded in this z. So your free energy, here it's Helmholtz free energy, because your work degrees of freedom are not there. So your free energy is related to the partition function by minus kBt times log of z. So you can have magnetic field, and your x can be like polarization or magnetization, or your or your j can be pressure itself. So your x is now a random variable, and your Hamiltonian will be changing now because you're adding this work degree of freedom. So your Hamiltonian will become h minus j dot x, and your probability distribution is just you change your Hamiltonian by h minus j dot x. Yeah. And this z is the Gibbs canonical partition function, and you can evaluate it in that way. And that is related to the Gibbs free energy of the system, because now you have the work degrees of freedom, and your g is equal to minus kBt times log z again. So these are the various ensembles. I have shown a picture. So I want to comment about entropy. So if you have a probability distribution, you can associate a increasing function with any probability distribution, and it can be denoted like this. So s equal to minus kb times log of f log f, which is like the average of log of f. So this s gives you how much disorder is present in the system, and what is the lack of information in the system. And when you apply this formula to all the ensembles, you get these formulas, and it's seen that these formulas are the same as the ones in thermodynamics. So your thermodynamic entropy and the statistical entropy matches. So now I come to molecular simulations. So so we saw for calculating averages in canonical ensemble, you do this, but this is only possible for very few cases. So it's not been done analytically for most of the systems which we have. They have a squared term in P, but if you don't know the potential, the integration becomes very tough. So we need to resort to numerical methods. And when we talk about numerical methods, we can do a brute force numerical quadrature, but this is very computationally expensive. So you will need, it goes as order of m power m, where m is the number of steps in the real line for integration, and n is the number of particles in the system. So for resolving this, there are two techniques which are largely used for molecular simulations. One is the Monte Carlo technique, which samples only those points in the phase space which have large probability and neglects all the others, and average is computed with this sampled points. And the other is the molecular dynamic simulation, which I'm going to talk about more, which finds it time. It samples the phase space in time, and you calculate time averages using this molecular dynamics. So I'll talk about molecular dynamics and the main steps in it. So these are the 4 steps in this molecular dynamic simulation. So first one is system preparation, then minimization, and then equilibration and production. So for system preparation, you want to, so you have a system to simulate, and you want to build your system. You want to assign your force fields, and so by force field I mean the interactions and your forces, and yeah, so these are the 2 parts which are necessary. So so what molecular dynamics does is do Newton's law, use Newton's law, and calculate the trajectories of each degree of freedom present in the simulating mixture. So if you have, so you need to assign a force field which will calculate the amount of forces at each time step, and some common force fields are are there in literature, and it includes terms like non-bonded interactions, which have like Coulombic interactions, Lennard-Jones potentials, and also bonded interactions, which means the chemical bonds, and they are usually represented by harmonic potentials, and it can be in bond vibrations, angular vibrations, and torsional vibrations. So one more thing which we want to do is apply periodic boundary conditions to remove finite size effects. So when you are simulating, typically you will have around 1000 atoms, and this will imply that your edge effects are very important. So you want to decrease your edge effects, or else it will perturb your system in ways which we don't want. So for this, you do a periodic boundary conditions, which looks like this. So you do the same copy of the system a lot of times, and you put cutoffs so that one system doesn't interact with others, and you get a bulk-like simulation. The next step is minimization, which means you would not want your system to initially blow up. So you are randomly, for example, if you have a solvent, you are randomly assigning positions to the solvent molecules. But if they are very close together, and if you chose a force field which goes as suppose 1 by r, it would blow up if they are very close together, and we don't want them to happen. So we will find a local energy minima near those near the system prepared thing, and by this process you will assign a state which can be the start of the simulation. So minimization just gives the positions of all the particles present in the simulation. So you want to assign velocities also, so that you can apply Newton's laws, and for that you can sample velocities from the Maxwell's-Boltzmann distribution, and you can and if you assign that, and you should keep into mind that your center of mass velocity should be 0, because your system box has to remain at the same position. So you usually subtract that system the center of mass velocity to keep your center of mass velocity 0. The next step is equilibration, where you want to so what is done in molecular dynamics is you want to reach a certain macroscopic state, for example N, V, and E, from an initial state. So for reaching that, you will need to equilibrate your system, and if you want to do this, you have to run the system in and wait for it, so that your macroscopic properties remain constant in time. They will be fluctuating because it's a small system, but on average it should remain constant in time, and you should continue doing equilibration till then. While doing the simulation, there are two parts. The first part is the force calculation at each time step. So this is the most computationally intensive step of molecular dynamics, and if you take pairwise interactions, the complexity goes as of n square, and different approximation in the force fields which have been coming from lot of research reduces this time complexity. After evaluating these forces, you need to integrate the motion of all the particles in a small time step. For doing that, you can use integrators, and they can be both deterministic or stochastic. Most of the deterministic integrators use this algorithm, where you find the positions at t plus delta t from the positions at t and the positions at t minus delta t, and the forces evaluated at t. So by this you can find the position of the system in the next time. So once you have completed equilibration, you have reached your target ensemble properties of NVE or NVT, And once you have reached that, you can start collecting data from this. And by data, I mean you can, for example, calculate temperatures, pressures, and all sorts of macroscopic averages. You can calculate free energies, and so on. So production always needs to be preceded by equilibration, so that the measured quantities are accurate. Now, this Verlet algorithm, which I told you, this conserves energy, because it's like a Newton's law type algorithm, and that is known for conserving energy. But if you want to simulate your ensemble in a canonical, if you want to simulate your system in a canonical ensemble where you have constant temperature, you would need other degrees of freedom to come in, which gives the constant temperature. So this is achieved by coupling your system to a thermostat. And there are various thermostatic algorithms which we need to choose so that we keep our temperature constant. And by temperature, I mean in a MD simulation, it is got from the equipartition theorem, which says the average kinetic energy is the number of degrees of freedom times half kBT. So I list down some popular thermostats over here. One of them is simple velocity rescaling, which evaluates the temperature at an instant and rescales each velocity by the ratio of the temperature to the temperature which you want. But this is very unphysical because it doesn't happen anywhere. The another thing which is done is Anderson thermostat, which works on a stochastic principle. So this actually chooses a number of particles in the ensemble and does random collisions with them and changes these. By random collisions, I mean assigning random velocities sampled by the Maxwell-Boltzmann distribution. And it is seen that this gives the canonical ensemble. But this should not be applied to systems where you want to evaluate the structures, because the dynamic properties of the system can be affected a lot by these abrupt collisions. This is the most popular algorithm of thermostats. One of them, how it works is assigning an additional degree of freedom to the Hamiltonian, the system Hamiltonian, which is S, and the momentum corresponding to that is. These are the extra terms. And when you apply these terms, it can be shown that when you calculate average with this Hamiltonian, you get and you do it in a microcanonical ensemble with this Hamiltonian, it's the same as the canonical ensemble with our original momentum and positions. So usually our proteins and all such stuff are in a constant pressure environment and a constant temperature environment. So we would ideally want to do a NPT ensemble simulation. So for this, we need to control the pressure also. So we use barostats for this purpose, and you have similar algorithms for barostats as thermostats, and you use these for doing the NPT ensemble. Now I would like to comment on equilibration workflows. So if you want your production ensemble to be in a microcanonical ensemble, it is suggested to first do a NVT ensemble simulation, and then so that it reaches equilibrium. This is helpful because NVT can couple to a heat reservoir, and it can take energy and lose energy and slowly, reach equilibrium quickly. And if you have an NVT ensemble, then you can just directly do it if you know the density. And if you don't know, you couple it to a NPT ensemble, find the density, and then go to NVT ensemble again. And if you want to do a NPT ensemble in the production, you first do a NVT where you go to the temperature, the temperature of interest, then do NPT to go to your pressure of interest, and then you start producing your data. So you should question yourself before running a MD simulation if it is actually required, or should you actually run an MD. So you should have a count of your computational resources which you have, and how much compute resources on average this simulation would take. So the time step which is used for integration, you need to set a time step which needs to be like 1/10 of the fastest vibrational frequency of the vibrational motion present in the system. If you take it as 1/10, then only that Verlet algorithm which I told you about, it conserves energy, or else it doesn't conserve energy. So your time step is limited by the highest frequency motion in your system. And you should know the time scales of the process which you are examining. For example, if you are seeing a conformational change in a protein, you should know how much time that usually takes. And if you divide that time by your time step, and you get to know how much, you get to know how many time steps you need, you can get to know how much compute power you require. And most of the simulations now, the maximum which has been achieved are in microseconds to millisecond simulation time. So if your system has a larger time scale than that, it's not wise to go for an MD, because you would not have resources to take care of it. Things to take care of when you're doing an MD. So first you take a stock of your plans. So by that I mean you see what your system is, you choose your force field, you choose your ensemble, and so on. Then you prepare to implement them. So you choose an appropriate software package. For example, you can use GrowMax or something, and you check if your force field values are actually accurate to your system. So I haven't talked about cutoffs a lot, because so when you do a periodic boundary condition simulation, you will have to make your system size large enough so that your non-bonded interactions don't cross that. So you can cut it off inside that simulation box, or else your non-bonded interactions might interact with its copies, and which we don't want. So you do your cutoffs properly. Then you choose appropriate settings for your desired ensemble. If you are doing it in a canonical ensemble, then you need to choose a proper thermostat. If you are doing it in a NPT ensemble, you need to choose a proper barostat, and so on. Then choose an appropriate time step. So as I told, you should choose less than 1/10 of the fastest vibrational motion present in the system. And then determine your run protocol. So I showed you for equilibration there are many protocols, and you can choose your thing which suits you the best. So one thing to keep in mind is, though we are simulating a system with all microscopic laws, your MD doesn't provide all the microscopic mechanistic detail of what your process is happening. Instead, what it does, it provides an average, or so you should not this is the last step. So we should not do MD by thinking that we will get all the detail of the system. Instead, we can measure average quantities and so on. So we should not use all the data that is generated, because if we use all the data, we might assume we have discovered something new, but which might not be the case. So in conclusion, I would like to say molecular dynamics are very exciting, and they provide some mechanistic detail of what is happening. It plays a significant role in our field. It gives new experimental ideas, and it directs experiments and so on. And advances in hardware, software, and methods can make these MD-based calculations particularly appealing for molecular design, and so simulations can guide your experiments, and you can precisely compute thermodynamic and statistical properties of these systems. Yeah, so I would conclude here. These are the references. Yeah, thank you, and I'm open for questions.
We will be having the next session now, so if you have any questions, you can just mail it to our mail ID, which is there in the brochure.
Savam, I can start. Thanks to the ISC software team, I'm sure everyone had an opportunity to learn a lot. Now we have a talk by Mr. Dhruv Gupta. He is a fourth year undergraduate at ISC. He'll be talking about genetic engineering, combining genetic circuits with the principles of VLSI to arrive at biobricks. He's also the president of the JRD Tata Innovation Support Center at ISC. And yeah, I'm sure we'll all get to learn a lot from the talk.
Sure.
This is surprisingly difficult to put on. Can you guys hear me? Hi. Am I audible? Thanks. Before starting, I would like to get to know a little bit about you guys. What all teams do we have here? Teams from which colleges? You guys. IIT Bombay. There. IIT Madras. Avandat bhai. You're the one from IIT Madras. तो तुम कौन हो? नहीं तुमको पता है यार तेरे पीछे वाले. IITBM. You guys? ISC. You? MIT Manipal. Nice range. The ones in the middle. I know you. UG here. All right, all right. So a lot of ISC students. Great. How have the previous sessions been? Why don't you tell me? It's been good. General question. What do you guys think AIIM is about? IIT Bombay guys. What is synthetic biology? Not you. Those. IITBM.
Like modifying something which is not present, messing up with nature basically, but in a good way.
Can you try to? So where does the concept of biobricks or genetic circuits fit in?
Genetic circuits and biobricks. From the point of our perspective, our view is like we are trying to introduce few genes in a bacteria to engineer to metamolecular engineering to produce a plant secondary metabolite.
Okay, so necessarily in a bacteria, can we also have in vitro solutions?
Yeah.
So what is synthetic biology? So all right, genetic circuits and introducing certain capabilities in organisms to allow robotic output, essentially which is function oriented and not survival oriented, seems to be one of the goals of synthetic biology. Anybody has something different to say? You guys. passing the mic. Yeah. The question was genetic circuits and essentially creating robotic life forms.
Oh,
So
you
yeah.
can use genetic circuits in a lot of stuff, but I'm speaking from like one of the projects we were looking into. But we wanted to replicate JK flip-flops in order to basically perform a certain function to convert very specific amino acid residues into some other residues by producing enzymes only at that point in time. And that was very, very nice because
Yeah.
it gave us the opportunity to be able to control which one of those residues we can choose and which one of them we don't want to, hydrolyze or whatever, perform certain functions on it. So
Alright.
it was, yeah, that was nice.
No, that's quite interesting. So since you mentioned JK flip-flops, that means you're familiar with VLSI. Is that
I am not extremely familiar, but there was people in my team who were from biomedical. That way we had somebody to guide us through the understanding of what was going on. We practically had to learn everything from scratch, but we do,
Great.
but a little bit of an idea is
right.
there.
So flip-flops are a digital circuit component, right, which are used for storing information. And you attempted to create one through a DNA-based substrate, or in your case, an amino acid-based substrate frame. This talk is very similar. We are using VLSI principles, analog circuitry, to create neural networks on a DNA frame. I will use my slides like this. Don't expect me to go into the presentation. Who knows about neural networks? Anybody? Yeah, of course. Go on. Mike. Somebody give this man a shield. No, a mic.
So neural networks are one of the essentially algorithms used for machine learning, mainly in supervised learning, and one of the most famous examples of that is the MNIST dataset training example that is given to beginners for
Right.
neural networks.
So neural networks, machine learning is about labeling data and sorting it. It's about pattern recognition. Some of the oldest neural networks are winner or loser takes all neural networks. Essentially where you have a set of competing neurons who each fire when you're given an input. In this example, you have the inputs being 1, 4 and 8, and the neurons corresponding to it fire with certain values when they receive 1 as an input, 4 as an input, or 8 as an input. You can see these values represented here. So say for example, when the actual class, when 1 is fired, when 1 is input, the neurons corresponding, so the 148 neurons, 1 gives 623 as a result, 0, 5 and 2. This circuit essentially floors 0 and 5 and 2 both to 0. The winner, 623, takes all. That is the output. So essentially this is how they recognize patterns. Anything which exceeds both the other values, the one for which recognition is more than competing values, takes all is the output, and the other values are ignored. Loser takes all does the opposite thing. The one for which there is lowest recognition, that is the one which fires the most. So essentially in this case, if 1 is output, it tells you the output is definitely not 4. The input is definitely not 4. That is what a loser takes all circuit does. Right. Now, surprisingly, okay, just a basic conversion from winner take all to loser take all. If you have x1, x2, x3 as winner take all inputs, you will receive, say if they are in ascending order, x3 being the largest, then x3 will be the output. Essentially, this is the only output neuron which will fire, the others won't. In this case, essentially in a loser take all circuit, you have the winner takes all circuit in the middle, only that x1 would produce two entities, essentially y2, y3. x2 would produce y1 and y3x. These are placeholders for the same, but they're producing the analogous quantities. So their competing quantities get amplified by the smallest input. So essentially, the smaller the input, the more the competing entities would be produced. And hence, then if you implement a winner takes all circuit, the competing entity, the smallest input which produce the most competing entities would be the output. Surprisingly, you can, since you talked about digital circuits, right, where we work with binary values, Digital logic involves computation with 0 and 1. You make logic decisions, you use AND, OR, and NOT to arrive at computations. Analog circuits harness the basic physics of a circuit to compute. For example, in an RC circuit, which you would all studied in high school, 11th, 12th grade, RC circuits, you have a resistor and a capacitor. The input and output, the voltage current correlation is a differential equation. That means with a simple analog circuit, you can solve differential equations, something which requires a lot of circuitry in digital architecture to do. So analog circuits are powerful, but they are noisy. Sound similar to generic circuits. So in this case, I will not go into the details of this because I will definitely lose the attention of everybody here. But how a winner takes all analog circuit works is, you have transistor gates. So these are current conveyors, which each mimic the input current. So if this is I1, which is the input, this belt would also mimic I1 in the output belt. And say that inputs are arranged in the increasing order. What would take place is that each of these inputs is biasing this transistor. And they're all connected to the same voltage gate. So the smallest current would bias the corresponding transistor the least, and is the most likely to switch off because they are all being held to the same reference value. So only the transistor corresponding to the largest current would switch on, rest every transistor would switch off. Winner takes all. The largest current takes all. The concept being annihilation and competition. The input currents are competing, annihilating each other until one input current is left, which is the output of the circuit. Somebody decided to make a genetic circuit which does the very same thing. Alright, a DNA can provide frame for an analog circuit. Now this diagram doesn't really tell much. Let us see what exactly goes on. So this reaction takes place in a thermocycler. In a standard thermocycler, you've got a bunch of DNA residues being recycled. Takes place at 37 degrees magnesium bath for stabilizing DNA. All standard. 3 input strands, X1, X2, X3 come in. Right. So you want to compare which one of X1, X2 or X3 is the one in the lowest quantity. That is what you want as output. You have gate complexes, which essentially gate complex G13 and G12 would both bind X1. So X1 for denotion and produce Y2, Y3. Same as we discussed in the initial neural networks case. So if X1 is the least, right, the production of Y2 and Y3 would be the least, but if Y3 is the most, production of X1, essentially X1 which corresponds to Y1, increases the most. This essentially does so this is the same as taking average ka -1. You average the 3 things out and you subtract the same quantity from it, right? So you subtract it from the average, so that means you reverse the inputs, input reversal. This is what the gate complexes do. Stand neutralization. So once you've got the reversed input, so if X1 was the least, Y1 becomes the most. If Y3 was the most, Y3 becomes the least. These reverse inputs are taken, and there are annihilator complexes. What the annihilator complexes do is that NiH12 would bind both Y1 and Y2. Essentially, it's a place for a neutralization reaction, where Y1 and Y2 would form a ternary complex, neutralize each other, and displace out of the reaction. So essentially, every single time a strand comes to an annihilator strand, they would compete with each other and would annihilate until the one with more quantity is the only one left. This takes place pairwise on 3 strands, Ni12, Ni23, and Ni31. So if X1 was the least, Y1 would be the most and would be the only reactant left at the end of this cycle. And that is what you obtain as the output, which is then amplified through PCR, and you obtain, you can understand that, yes, X1 was the least input. science runs a neural network on DNA frame. This talk was essentially about systems engineering. You have three systems: computation, computer science, VLSI, and synthetic biology. Being analogous to each other, unke concepts, the framework remains the same. The details change, the details of implementation, the specifics. The way that implementation of annihilation is done in a VLSI framework is different from how it is done in the genetic framework or the computer science framework. But the concepts of input reversal, of annihilation, computation, and output conversion are the same in all three frameworks. This is a principle because in engineering, in the field of computer science and VLSI, a lot of things that we want to solve for, any robotic function that we might want to perform, have already been solved. They're all solved problems. Hence, taking in framework inspiration from the same is a good idea. You work with a system which has already solved that sort of problem. You utilize its principles, not the specifics, the principles. You create a framework around it, and you apply it to the problem that you want to solve. That will be all. Thank you. Thanks, Shubham. Take my laptop and we'll return your mic to you.
We are serving tea and coffee outside right now.